Welcome back to the Deep Dive, where we extract the most impactful insights from the cutting edge of science. And today,
Oh,
we are skipping the gentle introductions. We are diving, headfirst into the biggest cold case in history.
the biggest one.
Where did we come from? And not the nice, easy story you might remember, not some, peaceful, sun-drenched puddle.
No, this is the opposite of that. We're talking about the absolute terrifying, forgotten bottom of the ancient ocean.
That's right. We're going miles beneath the surface where the pressure is crushing, the light is, that's non-existent.
And the only energy source is this primal chemical fury of rock meeting water. We're asking where the heck life took its first breath.
So our mission today, based on our reading, is to really examine this radical theory.
Uh-huh.
The idea that life didn't kick off in Charles Darwin's famed warm little pond, but in the dark, alkaline depths of hydrothermal vents.
And our research gives us the goods on this. We have concrete lab experiments that prove it's chemically plausible.
And it drags us directly into the modern hunt for alien life. Specifically on places like Jupiter's ice-covered moon, Europa.
Oh, yeah, and we are going to get into the nitty-gritty of that chemistry. You're going to learn about a hidden underwater metropolis that's existed for, 100,000 years.
It's 100,000.
Yeah, which makes it an ideal cradle for slow, deliberate evolution. We're also going to look at how scientists are successfully manufacturing the actual building blocks of life, amino acids, using a cheap, abundant mineral called green rust in a simple laboratory beaker.
Wow. Okay, but plus we have to talk about the brutal reality check. There's a critical piece of chemistry that might just, melt the whole hot start origin story entirely.
The RNA problem.
The instability of RNA and why temperature is its absolute enemy. We'll break down exactly why functional genetic material just falls apart in seconds if it gets too hot.
And of course, the alien connection. We found this story of a completely isolated, single-species ecosystem thriving 2.8 kilometers deep.
Wait, inside a gold mine?
Inside a South African gold mine, powered by core radiation. And that weird little microbe might be the perfect blueprint for life thriving right now, independently, on Europa.
Okay, so we're framing this as the ultimate origin story showdown.
For sure.
To set the stage, we have to recognize that historically, the search for life's origin has been defined by two completely different environments.
Absolutely. The first one, the classic model, is the warm little pond, or, primordial soup.
The one we all learned in school.
Exactly. And this requires solar input. It's a photosynthesis-based scenario where energy comes from the sun, and the concentration of organic materials happens in shallow pools or terrestrial settings. It's a surface-based, sun-dependent start.
It's the comfortable narrative, right? You have lightning, UV radiation, gentle cycles, and things just slowly accumulate under the warmth of the sun.
Then you have the Challenger, which only really gained traction after deep-sea exploration took off in the 1970s.
The deep-sea hydrothermal vent.
Right. And this proposes a chemototrophic origin.
Chemo.
Chemototrophic. It just means life dependent solely on chemical energy derived from the reaction between rock and water, completely independent of the surface or sunlight.
And the reason this theory is so fundamentally important is that it just changed the rules for astrobiology, didn't it?
You're completely right.
If you don't need the sun, the habitable zone of the universe just became massive.
Precisely. The whole shift in astrobiology to prioritize moons with subsurface oceans like Europa or Enceladus, that's a direct result of the vent theory, proving life doesn't need external sunlight. It just needs sustained chemical gradients.
So was the birth of life a gentle, sunlit cradle or a high-pressure subterranean chemical reactor?
That's the question.
Let's jump into the abyss and start our deep dive. All right, let's start at the bottom of the ocean.
Let's do it.
When we talk about hydrothermal vents, I think most people picture those aggressive, superheated black smokers. These giant smoking chimneys just spewing black plumes of sulfide mineral.
Yeah, they're dramatic.
They are. But why, despite hosting these incredibly robust, modern ecosystems, are the black smokers likely not where life originated?
Well, the black smokers are geological beasts. They are high temperature, sometimes over 350 degrees Celsius.
That's insanely hot.
It's incredibly hot. They're also highly acidic and just saturated with toxic metals and sulfur compounds. So while life adapts to them beautifully today, trying to originate the delicate, complex chemistry of a cell in that environment,
This is too much.
it's incredibly difficult. It's too transient, too hot, and too acidic for early molecular stability.
So you're trying to build the foundational genetic material material while it's being simultaneously boiled and pickled.
Yeah, that's a good way to put it. It's a really unstable environment for a genesis event.
So what's the alternative?
The real candidate, the dark horse theory, it centers on alkaline hydrothermal vents. And the crown jewel of this hypothesis is the Lost City hydrothermal field,
Lost City.
discovered on the Atlantis Massif at the Mid-Atlantic Ridge.
The name, Lost City, immediately captures the imagination. It sounds like something out of a movie.
It really does.
Our research highlights that it was only identified in 2000, and its significance comes from its incredible longevity.
And this is the key. This is a geological time capsule. Lost City is estimated to be older than 120,000 years.
Wow.
Now, to put that in perspective, the black smokers we've studied, they're often active for mere decades, maybe a few hundred years at most.
So Lost City has been active for 100 times longer than the average black smoker.
At least.
That longevity is just.
Yeah.
It has to be key. Evolution is not an instant event. It needs stable conditions over immense spans of time.
It does. A geological cradle that persists for millennia offers the necessary stability for slow, incremental assembly of complex systems.
And what do these things look like?
The architecture is stunning. The structures are these massive carbonate chimneys, chalky white, almost ghostly.
Not black.
Not black at all. The largest one, they nicknamed it Poseidon, and it towers 60 meters tall.
That's a skyscraper.
It is. And these aren't volcanic rock. They're formed because hot alkaline fluids meet cold, acidic seawater.
And that causes what?
It causes calcium carbonate to precipitate out of the solution, building these towers slowly, molecule by molecule, over vast amounts of time.
So let's get into the chemical contrast. What is Lost City spitting out that's so different from the corrosive black smokers?
These are known as non-smokers.
Okay.
They don't produce much carbon dioxide, sulfide, or heavy metals. Their fluids are relatively mild, low temperature, ranging between 40 and 90 degrees Celsius.
So warm bathwater to hot tub temperature.
Exactly. And critically, they are highly alkaline, with a pH over 9.
That high pH is our first clue to the energy system, which I know we'll unpack. But the actual chemical emissions, that's what matters most.
It is. And what they're emitting is methane, CH4, and large amounts of diatomic hydrogen, H2.
Hydrogen gas.
Lots of it. And that leads us directly to the engine room. Serpentinization.
Serpentinization. It sounds complicated.
It does, but this is the key geochemical process driving this whole system. It's the foundation of the deep-sea origin theory. And really, it's complicated in name only. It's essentially the simplest, most reliable form of natural chemistry on Earth.
Okay, so let's break down serpentinization, because it sounds esoteric, but you're saying it happens everywhere there's rock and water.
Correct. Serpentinization occurs when ferromagnesian silicate minerals, specifically olivine and pyroxenes, come into contact with water deep in the crust.
And the place where Lost City is, the Atlantis Massif, is that special.
It is. It's composed of ultramafic rock, which is just dense in these specific iron and magnesium-rich minerals.
So the rock itself is unusually rich in the exact ingredient needed for this reaction.
Exactly. When water penetrates these minerals, it causes them to hydrolyze. The water molecules break down and react with the iron in the rock.
What happens to the iron?
This reaction converts the iron, it oxidizes it, which in turn reduces the water molecule.
And what do you get when you reduce water?
You get molecular hydrogen gas, H2, and methane, CH4, along with the heat that makes the reaction exothermic. The process itself fundamentally rearranges the composition of the rock, turning olivine into serpentine minerals.
Hence the name.
Hence the name, but the byproduct is the critical energy molecule, H2.
So H2 is the fundamental fuel. Life didn't need sunlight. It just needed rock chemistry. This hydrogen acts as the reducing power necessary to convert inorganic carbon dioxide into usable organic molecules.
It's the geological battery we mentioned.
It's more than a battery, though, isn't it?
It's more than a battery. It's a non-stop, self-regenerating fuel cell. This constant supply of hydrogen has led to the compelling theory that the last universal common ancestor, LUCA, of all life on Earth, was likely a chematotroph. It may have depended entirely on the geochemically sustained proton gradients in H2 produced by these serpentinization systems.
That is a wild thought. It means every single thing alive today, every bacterium, every tree, every complex animal is descended from something that essentially ate rock and drank hydrogen.
That's the idea.
That's a fundamentally different Genesis narrative than a sunny pond. It means life started in the maintenance room, not the lounge.
And it suggests that the core metabolism of early life, the very first steps toward energy production, were dictated by the geology of the ocean floor.
If the genesis point was alkaline, hydrogen-fueled, and rock-sustained, then we have a very strong alternative to Darwin's original hypothesis.
So we have the energy source, the rock generating H2.
Right.
Now we need to ask, how did that H2 turn into energy that a protocell could actually use?
Yeah, that's the big leap.
This is the issue of chemiosmotic coupling, which is the universal energy currency of life.
You hear that phrase, and it sounds like a textbook nightmare. But can you explain it simply?
Yeah.
How does a single cell power itself?
Nearly all cells, from the simplest archaea to our own mitochondria, use an electrochemical proton gradient across a membrane to synthesize ATP.
ATP, adenosine triphosphate.
The high-energy molecule that fuels all cellular processes. Protons, which are just H+ ions, are pumped across a barrier, creating a concentration difference, a gradient. And then, when they rush back across that barrier, they spin a tiny molecular turbine, ATP synthase, which generates ATP.
So it's like building up water pressure behind a dam and then letting the water rush out to turn a turbine and generate electricity?
Exactly. The pressure is the proton gradient.
That's a great analogy.
Now, this mechanism is so complex and universal that it must have emerged very early. But how did the very first primitive protocells, which lacked the necessary protein pumps to create their own gradient, how did they manage this?
That's the problem. Evolving a protein pump and a turbine at the same time in the first cell seems like an impossible molecular leap, a real chicken and egg problem.
It does. But this is where the alkaline vent theory solves the problem beautifully. The vents naturally provide the gradient. As we established, the vent fluids are highly alkaline.
Low proton concentration, pH 9+.
And the surrounding ocean water is mildly acidic, higher proton concentration.
So the moment those two fluids meet inside the porous chimney structures, the difference in pH automatically creates a proton pressure gradient.
Yes. The micropores within the inorganic chimney walls act as the primitive membrane.
The rock is the membrane.
The rock is the membrane, and the difference in pH between the alkaline vent water inside the pores and the slightly acidic early ocean water outside means that H+ ions are constantly trying to rush into the pores.
It's a freebie.
This natural, geologically sustained proton gradient replaces the need for the cell to spend energy pumping protons. The rock acts as the first power source and the first membrane.
That's incredibly elegant. Life didn't have to invent the energy system. It just had to hijack the one that geology was already providing for free.
It saves eons of evolutionary time.
It must.
And we can't forget the catalysis. Within these inorganic barriers, we find catalytic mineral surfaces.
So not just a membrane, but also a workshop.
Exactly. We are talking about the iron-sulfur world hypothesis. Specifically, minerals like nickel-doped maconite, which are naturally formed in these alkaline conditions.
That sounds like a heavy metal band name.
It does, but it's critical chemistry. These mineral structures are chemically analogous to the iron-sulfur cofactors found in many modern metabolic enzymes, particularly those involved in fixing carbon.
So the vent environment didn't just provide the energy gradient.
It provided the inorganic machinery to facilitate the first metabolic steps, predating the need for fragile organic enzymes.
Okay, so we have the energy and the catalysts, but we still need the actual molecules that form life, the carbon backbones. How did we get organic compounds from hydrogen gas and dissolved CO2?
That's achieved through abiotic organic synthesis, specifically Fischer-Tropsch type, or FTT reactions.
FTT.
This is where the continuous supply of H2 and CO2 from the vent fluids in the early ocean comes into play.
I remember FTT from high school chemistry, but give us the easy version.
Okay, so FTT processes are standard industrial chemistry hijacked by nature. They use metal catalysts in the vent context, iron, nickel, or their oxides, like the magnetite formed during serpentinization, to convert carbon monoxide or carbon dioxide and hydrogen gas into reduced carbon molecules. Industrially, FTT is used to convert coal or natural gas into liquid fuels today.
So it's a method for making big molecules from simple gases.
Precisely.
So if nature runs the same process in the chimney pores, what is the crucial output for early life?
The output is linear hydrocarbons and, more importantly, linear fatty acids. This is the key linear advantage.
Linear, as in straight lines.
Straight lines. Think about a cell membrane. It's a bilayer made of long, straight molecules, fatty acids. FTT reactions preferentially produce those long, straight chains abiotically?
Meaning the required building blocks for the cell wall spontaneously generate within the Venn structure.
Exactly.
As a comprehensive self-contained system, you have the energy, the catalyst, and the raw materials for the membrane, all provided by the geochemistry of the Earth's mantle. But did anyone actually prove that this synthesis works under these mild alkaline conditions?
Yes, and this is where the empirical support gets fantastic. A team at NASA's Jet Propulsion Laboratory set up a simple electrochemical reactor to precisely simulate alkaline vent conditions.
Okay, so let's talk methodology. What did they actually put in the beaker?
They simulated the primordial ocean sow, water, minerals, and basic precursor molecules like ammonia and pyruvate. They heated it to a Lost City-appropriate 70 degrees Celsius,
Right.
adjusted the pH to be highly alkaline, and maintained a low-oxygen environment consistent with the early Earth.
And the chemical trigger, what was the secret sauce?
The essential ingredient was iron hydroxide, more commonly known as green rust.
Green rust.
Green rust was incredibly abundant on the early Earth because of the sheer quantity of iron in the crust. And the scientists found that when this green rust reacted with trace amounts of oxygen they introduced, the synthesis started.
What was the aha moment for the researchers? What did they successfully generate?
They successfully produced the amino acid alanine and the alpha-hydroxy acid lactate. Alanine is one of the foundational 20 amino acids that make up all proteins, and lactate is a versatile precursor to complex sugars and other essential organic molecules.
That is powerful evidence. It proves that the geological environment of these alkaline deep-sea vents is inherently conducive to synthesizing the foundational building blocks of life under mild conditions.
No lightning strikes or massive UV radiation required.
Right. But we do have to acknowledge the scientific skepticism.
We do.
Even with the successful synthesis of simple building blocks, the debate heats up when we look at more complex intermediary molecules.
Right, the molecules required to transition from simple amino acids to functional peptides or RNA.
Like what?
Our reading notes the challenge posed by molecules like acetylthioesters.
Acetylthioesters.
These molecules are thought to be necessary for bridging early metabolism toward modern life, but many researchers reject the idea that they could have accumulated abiotically in vent fields to any significant extent. They're just chemically unstable. The heat and the constant flow might break them down faster than they can form.
So it's a question of stability versus availability. We have the perfect environment for generation, but maybe not the perfect environment for accumulation and stabilization of the next crucial steps. Okay, that instability issue gives us the perfect transition to the cold reality check.
Yep.
If the deep-sea alkaline vents are so geochemically perfect, free energy, free catalysts, free linear fatty acids, why do the warm little pond advocates still hold such a strong position?
Well, it all comes down to the most sensitive piece of the puzzle, RNA.
RNA.
The existence of an RNA world where RNA acted as both the information storage like DNA and the catalyst like proteins, that is an almost universal hypothesis for how life started.
Right. It had to do both jobs at first.
But this is the powerful biochemical argument against the hot start origin. RNA and its building blocks are fundamentally thermolable. They melt.
And we need specifics here, because just saying it melts doesn't quite hit home. Give us the numbers our research provides on exactly how fast RNA degrades under hot conditions.
The degradation rates are shocking. I mean, really shocking.
Okay.
Consider the RNA-based cytosine. At 100 degrees Celsius, the boiling point of water, a temperature easily achieved in a vent system cytosine has a half-life of just 19 days.
19 days. That means you lose half of your genetic instructional set in less than 3 weeks.
Exactly. You can't build a viable system if your entire blueprint is constantly self-destructing. That rate of degradation means no stable repository of information can be maintained.
It's like trying to build a sandcastle in the surf.
A perfect analogy, and that's just the individual bases.
Right.
If we look at a typical moderately sized functional RNA molecule, say 2,000 nucleotides long,
Okay.
it's expected to suffer one chemical break one instance of backbone cleavage every 26 seconds at 100 degrees C.
26 seconds. You simply cannot evolve complexity when the molecules required for reproduction and catalysis are shattering every half minute. Evolution requires stability over millions of years, not seconds.
And stability isn't just about preventing breaks, it's about function. For RNA to work as an enzyme, it must fold into a precise three-dimensional structure.
Right, the shape is everything.
The shape is everything, and this folding is temperature dependent.
So how do scientists even measure that folding?
They use techniques like circular dichroism measurements.
Which is a fancy way of saying.
It's basically just a fancy way of watching the molecule curl up. It measures how light interacts with the molecule structure. And these experiments show that the crucial folding capability decreases rapidly above about 105 degrees C.
35 degrees, that's not even that hot.
It's not. And for many sequences, functional folding is virtually non-existent above 70 degrees C.
That is the ultimate killer for the hot start theory. 70 degrees Celsius is standard operating temperature for the lost city vents, let alone the 350-degree black smokers. If the core genetic material can't even fold to function above body temperature, then the RNA stage had to happen somewhere cool.
The conclusion is inescapable. Regardless of where the raw chemistry began, the stage where self-replication and complex catalysis took hold, the RNA world, must have occurred under mesophilic or moderate temperature conditions.
So we swing back to the surface, we swing back to Darwin, writing his 1871 letter to Joseph Dalton Hooker, musing about a warm little pond with all sorts of ammonia and phosphoric salts.
Darwin vindicated.
So what is the modern, scientifically robust version of the cold start alternative?
The modern hypothesis suggests shallow terrestrial environments like hot springs, volcanic islands, or evaporative lakes.
Why there?
Because low temperatures meet specific chemical mechanics. The great advantage these environments offer is concentration and cycling.
Tell us about the mechanics of the wet-dry cycles. How does evaporation solve the RNA problem?
When water evaporates in a shallow pool, the precursor molecules, the individual nucleotides, amino acids, lipids, they're physically forced into close proximity. This concentration overcomes a fundamental chemical hurdle, entropy.
Right, life requires organization.
And spontaneous polymerization requires driving out water molecules. It's a dehydration reaction.
So drying the pond literally provides the activation energy needed to link simple molecules into long chains, the polymers.
Exactly. When the pond dries, the molecules link up. When it rains or the tide comes back, the water reintroduces hydration, and those newly formed polymers are freed to interact.
How effective is that?
Numerical models demonstrate that simple polymers like RNA could have appeared after just several of these cycles. You are constantly driving the necessary dehydration reactions required to link precursor molecules into complex polymers.
That's a fundamentally different energy source. The vent uses chemical energy from rock. The pond uses solar energy and physical forces of evaporation to drive complexity.
And the pond theory also solves the issue of cell formation much cleaner than the salty ocean vent.
The vesicle formation argument.
Right. Lipids spontaneously form stable, semi-permeable protocells, the precursors to biological cells, far better in freshwater environments than in the salty brines of the early ocean.
Salt is the enemy of protolife. Too much heat, RNA breaks. Too much salt, the delicate membranes collapse. Why the salt sensitivity?
Early protocells lacked the sophisticated ion pumps that modern cells use to regulate their internal chemistry. High salinity destabilizes these simple membranes, causing them to collapse or dissolve. A freshwater terrestrial environment provides the necessary stability.
And what about the surface chemistry? The role of mineral surfaces.
Clays, specifically, are incredible catalysts for the cold start.
Clays?
Yeah, minerals like montmorillonite act as templates. They physically attract and concentrate the precursors on their surfaces, increasing the local reaction rate exponentially.
Has this been shown in a lab?
Oh, yeah. Lab work has successfully demonstrated that peptides' short protein chains up to 55 amino acids long can be assembled simply by using these mineral surfaces as templates.
The fact that a simple wet-dry cycle combined with clay can assemble a 55 amino acid peptide, that changes the entire timeline we imagine for abiogenesis.
It means complexity can happen quickly on the surface, which is exactly what the RNA stability issue demands.
So we have two fiercely strong, data-driven positions. We have the deep-sea vent theory, providing a robust, stable, universal energy source for building simple blocks.
And we have the pond theory, providing the perfect, cool, cyclical environment for stabilizing and folding the most critical, complex molecule, RNA.
It's a real paradox.
It is.
So are we deep-sea vent junkies, or are we pond people? We'd love to hear your take on the origin of life. What do you think? Comment below and let us know your favorite theory.
And before we move on to hunting aliens, just a quick note. We are a small channel, and if you are enjoying this deep dive and appreciate the details, please consider liking and subscribing to our YouTube channel.
Your support really does help us keep exploring the weirdest parts of science. Now that we've established that life only needs rock, water, and chemistry, Let's go alien hunting.
So the discovery of chemotoptrophy, that life can exist solely on chemical energy derived from rock, that is arguably the biggest paradigm shift in astrobiology.
It's huge.
We don't need a sun, we just need a liquid ocean in contact with a rocky core.
And when you say that, one name immediately leaps into the spotlight. Europa, Jupiter's massive ice-encased moon.
Europa is the prime suspect for current extraterrestrial life. It's covered in an ice shell that's 12 to 17 kilometers thick.
Unbelievable.
But beneath that shield is almost certainly a deep, salty subsurface ocean.
What makes scientists so confident that a massive body of liquid water is trapped under all that ice? How do we know?
We have several lines of evidence, but the most compelling came from the Galileo spacecraft. When Galileo passed Europa, it detected fluctuations in the moon's magnetic field.
Not Jupiter's field.
No, this was an induced magnetic dipole, which means the moon had a highly conductive layer close to the surface.
And the easiest explanation for a highly conductive layer is
a massive global ocean of salty water. Salt water conducts electricity brilliantly.
so the sloshing of the salty water under the ice is generating its own little magnetic signature.
Exactly. And we also have visual evidence on the surface, like features called chaos terrains.
Chaos terrains.
These are vast areas where the surface ice looks fractured and jumbled, almost like icebergs that have been churned and refrozen. It implies the ice broke up and liquid water may have welled up from below.
And the colors on those chaos terrains suggest the water is far from pure H2O.
That's the chemical link. Features like Theramaculae exhibit these reddish colors that scientists attribute to hydrated salts.
So a brine.
Concentrated brine that has leaked through cracks and frozen, providing a window into the composition of that subsurface ocean.
Okay, so the ocean is there, and it's salty. But how does that water stay liquid when it's millions of miles from the sun? Where's the heat coming from?
The heat source is planetary dynamics. Tidal heating.
Odd.
Europa is locked in a massive gravitational tug-of-war with Jupiter and its neighboring moons. Jupiter's immense gravity constantly stretches and squeezes Europa as it orbits. This internal friction, or tidal flexing, generates heat.
We see this effect most dramatically on Io, Europa's sister moon,
Right, which is the most volcanically active body in the entire solar system.
because it's closer to Jupiter.
Exactly, the tidal forces are even more extreme there. Europa is further out, so it's less volcanically active, but the tidal heating still accounts for an estimated 86% of the moon's total internal heat, enough to maintain that liquid ocean.
And here is the linchpin that ties this moon back to our lost city research. For life to originate via serpentinization, the ocean must be in direct contact with the rocky core.
This is the crucial habitability constraint.
So what do we know about Europa's geology in that regard?
Europa's ocean is estimated to be 100 kilometers deep.
10 times deeper than our deepest trenches.
But modeling suggests that, unlike other icy moons like Ganymede, which may have a layer of high-pressure ice separating the water from the rock, Europa's liquid ocean is in direct, intimate contact with the rocky mantle, the seafloor below.
What that means.
It means the conditions for serpentinization water reacting with silicate rock are almost certainly met.
So if that contact is confirmed, we have established the conditions for a deep-sea chemotrophic origin story.
We have.
Let's map out the 3 potential energy pathways for European life.
Okay, pathway 1, volcanic activity. If the tidal heating is strong enough to cause localized volcanic vents, we would see hyperthermophilic ecosystems.
Hot ones.
Very hot. Organisms could rely on anaerobic reactions like sulfate reduction, similar to Earth's most extreme examples, like the Archaea Methanoporus candleri, which thrives at 122 degrees Celsius.
A high-energy, hot start.
Yep. Pathway 2, which is lower energy but more certain, serpentinization,
Lost city model.
applied to Europa. Even without active volcanism, the constant contact between water and the iron-rich silicates in the mantle will generate H2 gas and CH4.
Providing the energy for methanogenesis.
Exactly. This is the oldest proposed life pathway on Earth, and it's a near certainty on Europa if the rock-water contact exists.
Then pathway 3 is the truly unique one, the radiogenic chemosynthesis pathway.
The gold mine analogy.
Right.
This is the low-hanging fruit of life support. Radiation from the natural decay of heavy elements like uranium and thorium in Europa's core can split water molecules deep in the seafloor sediments.
Water radiolysis.
That's the process. And it generates highly reactive chemical compounds, including H2 gas.
So instead of tidal friction providing heat, the simple decay of radioactive material in the rock is providing the chemical fuel by splitting the water.
It's a purely internal energy source.
And we have an analog for this on Earth.
We have a perfect terrestrial analog. In 2006, scientists discovered the bacterium Candidatus desulfurutus audaxviator.
That's a mouthful.
It is. They found it 2.8 kilometers deep inside the Mponing gold mine in South Africa. This organism lives in total isolation from the surface.
Wow.
It is anaerobic, sulfate-reducing, and forms a single-species ecosystem powered entirely by the water radiolysis caused by uranium decay in the surrounding rock.
That is the ultimate extremophile. It's living proof that you can have a complex, self-sustaining ecosystem built on nothing but water, rock, and core radiation.
It is. If life is found on Europa, it could look exactly like that thing, a simple, deeply buried sulfate reducer that's never felt the sun.
That one discovery just completely redefines the habitable zone.
It does. It suggests the habitable zone is not just a distance from a star, it's any environment with rock, water, and sufficient internal physics or chemistry to drive a redox reaction.
So let's broaden the context based on our research. What else in the solar system has signs of this serpentinization engine?
Saturn's moon Enceladus is an excellent case study. Its geysers jet plumes into space that contain silica, nanoparticles, and organic molecules like acetylene.
And the silica is the key.
The presence of silica is a smoking gun. It confirms that the water is circulating, coming into contact with silicate rock, and being heated exactly as happens in terrestrial hydrothermal systems.
And Mars, our closest neighbor, which had massive surface water billions of years ago.
On Mars, the search is for fossilized life in ancient hydrothermal systems. We have evidence of both magmatic and radiogenic heating in Mars's past crust.
So it could have happened there too.
And even today, serpentinization remains a perfectly viable geochemical route for generating H2 and CH4 in the deep Martian subsurface, meaning that if life retreated underground as the surface dried, those rock-eating microbes could theoretically still be there.
The upshot is that our search for aliens has become profoundly more specific and exciting. We're no longer just scanning for planets that look like Earth.
No, we are actively preparing to search for the chemical signatures of the Lost City model on other worlds.
Which brings us to the critical mission ahead, the NASA Europa Clipper.
That's right.
Tell us what this mission, scheduled to arrive in the 2030s, is actually going to do to confirm these deep-sea life pathways.
The Clipper is a full suite of instruments designed to answer the rock-water question. The most critical instrument for our deep dive theory is the Europa Radar for Ice Sounding, or REASON.
An ice-penetrating radar system.
Correct.
What's the specific goal of that radar?
REASON will map the structure of the ice shell. Crucially, it will try to determine the depth of the ocean and, more importantly, whether there is a liquid layer at the very bottom, confirming that rock-water contact.
If they see evidence of mixing or deep circulation?
It strongly supports the viability of serpentinization.
So it's not just looking for life, it's looking for the engine that powers life.
Precisely. And another key instrument is the Mass Spectrometer for Planetary Exploration, or MASSPEX.
What's that for?
MASSPEX will fly through Europa's potential plumes, if they are active, and sniff out the chemical composition of the ice and gases being vented into space.
So if they detect high concentrations of hydrogen and methane, along with specific trace metals,
that's a direct confirmation of active serpentinization.
it is a chemical confirmation. Finding high levels of H2 and CH4, especially when combined with evidence of silicate rock alteration, would suggest that the geological process that fueled the earliest life on Earth is thriving on Europa right now.
It ties our oldest origin story directly to our search for alien life.
The amount of information we have gained from studying a little patch of rock in the Atlantic Ocean is fundamentally guiding our $5 billion space mission.
It's pretty amazing.
That's insane. So we've synthesized two competing, equally compelling origin stories today.
We have.
The deep-sea vent theory, powered by rock geochemistry, offers a sustained energy gradient across inorganic membranes.
Perfect for building simple, linear molecules.
And the warm little pond theory offers the moderate temperatures and concentration cycles required for the stability and crucial 3D folding of complex genetic material, like RNA.
The central conflict here is really a matter of chronology and stabilization. It's less about which environment existed and more about which conditions were sufficient to stabilize the transition from simple chemistry to self-replicating complexity.
Right. Did the molecules originate in the dark abyss and then move to the cooler surface? Or did they arise entirely in the surface ponds, driven by solar energy and evaporation cycles?
That's the billion-dollar question.
The relevance to you, the learner, is profound. This deep dive shows that our search for life is no longer limited to finding environments that look like Earth's surface.
Not at all.
By studying extremophiles and abiotic chemistry in places like the Lost City, we expand our view of biology to encompass unfamiliar metabolic strategies.
The ultimate takeaway is that if life is defined by its ability to utilize a chemical energy gradient, then the universe is vastly more habitable than we previously thought.
And this geological perspective is why we are investing so heavily in missions like the Europa Clipper.
If we can confirm the rock-water contact and sniff out those plumes, we will know exactly what geological processes on Earth pointed us toward life elsewhere.
It's the ultimate convergence of geology, chemistry, and biology. The core mechanics of the planet dictate the core mechanics of life.
Which brings us to our final provocative thought for you to mull over.
Lay it on us.
We know that all existing life stems from LUCA. We also know that if a brand new protocell spontaneously formed today, it would be instantly devoured or outcompeted by existing complex life. A protocell is just dinner for a modern bacterium.
That's the tragic reality of being born too late.
But here's the kicker. If these unique alkaline hydrogen-rich conditions are still ongoing in unexplored pockets of the Earth's oceanic crust, and they are,
Yeah.
could simple non-DNA-based life still be forming spontaneously right now? New life that is non-competing because it's so deep down, relying entirely on the iron-sulfur world and green rust for its existence.
A completely alien, non-DNA, brand new form of life existing right now, miles beneath our feet, that we simply haven't noticed yet because we're looking for something too specific.
Something too complex.
That's a deep dive and a scary thought for another day. Thank you for joining us.
Until then, keep asking the big questions.